Good day, everyone. My name is Mike Pennington, and I'm pleased to present to you some of our recent research regarding a group of peptides called plant defensins and their inhibitory effect on Kv1.3 channels. I'm pleased to present this to you, and I thank the organizers for the opportunity to present some of our research. On the first slide, what we can see here are plant defensins are a group of conserved peptides found in plants which have similar properties to animal defensins, being that they are highly disulfide constrained. They are typically characterized as having greater potency against fungi as opposed to antimicrobial effects against bacteria, which typically are the animal type defensins. They are typically peptides with links between 40 and 55 residues. And characteristically, they have a series of 4 nested disulfide bonds. These internal disulfides are very similar to those of several of the scorpion short toxins, which bind to the K channels on the peripheral membrane pore. And they basically serve like blocking the channel, in essence, like a cork in a bottle. And our speculation was perhaps these molecules, since they show high homology to some of these scorpion type toxins, may actually bind to Kv1.3 channels. What we can see, if we look at the 3-dimensional structure of several of these peptides and compare them to the short toxin called Agitoxin from scorpion venom, you can see that they have the characteristic 3D fold, which shows a high degree of homology to these types of molecules, and that being a 3-stranded anti-parallel beta-pleated sheet with a short stretch of alpha helix that you see in all of these molecules. And although we have worked with plant defensins from numerous different species, we're going to focus on 2 specific peptides that come from one isolated from the common grape called VVK, and the other one isolated from the African oil palm called EGK. The sequences of these peptides, VVK1 and EGK1, are shown in this table here. As you can see, both of these are approximately 47 residue peptides, all contain the same 4 disulfide bonds that we were discussing earlier. But interestingly, they have this internal motif located here, which is highly basic and very reminiscent of some of the cell-penetrating peptides, most notably peptides like TAT, which are known to translocate across the cell membrane. And we speculated that this could be potentially part of the mechanism of action which these peptides would have. In terms of the molecule themselves, you can see they're considerably longer than the alpha scorpion toxins. These are typically around 35 to 37 residues. Shown here, we show alpha KTX 3.1 and 2.2. These peptides are typically around 37 residues, like I said, and we tried to design one additional molecule that we called EGK6, which was truncated, eliminating this disulfide bond, which linked the C-terminal to the N-terminal portion of the molecule, trying to truncate that and give us a true scorpion length type toxin to see how that may potentially interact with the Kv1.3 channels. And so what I'm going to discuss with you are the peptides VVK1 and EGK1, and of course the truncated analog that we mentioned earlier. We also prepared an optimized sequence here, EGK5, and a TAMR labeled material, which was set up to help us determine whether the peptide in fact would translocate across the membrane. The interesting thing with regard to the design analog is we based this upon molecular dynamic comparisons between EGK and VVK. And we selected the substitutions of Q6A, K11E, R16G, A17D, R35K, and F37L. We made these to establish the same type of ionic interactions that were occurring in the peptide with the molecule E11 to R40 and D17 to cis34. The Q6A change was made to eliminate the R40 side chain switching from E11 to Q6. The R16G, R35K, and F37L were made to make the peptide more EGK-like because these now predicted new ionic bonds and they potentially may have a more stable overall structure. The next slide, we see our overall strategy to produce these peptides. In this particular slide, I focus on only VVK1, but the same type strategy was used for all of the molecules that we made. We selected a low substitution chem matrix resin, which is ideally set up for making longer, more complex peptides such as VVK. We also included key pseudoproline derivatives to help minimize resin-based aggregation, which can occur on longer peptides, as well as a dipeptide to eliminate tough to remove impurities that you see there, the GG. The C-terminal cis acid, we were worrying specifically about its possibility of racemizing. In fact, you will see that we did observe some of that in terms of an impurity that was generated. And also we know that we have a very tough ARG-rich cationic section, which will present some difficulty as TAT-related type peptides are all quite difficult to make an FMOC synthesis. For the 4-disulfide bonds, we selected cis-tridal and would use God-assisted folding, as I like to call it, hoping that we get the ideal thermodynamic isomer forming, which is the biologically favored species. Following preparative HPLC, the linear crude peptide was then subsequently folded in the conditions that you see here, 0.1 mg per mil in 50 millimolar ammonium acetate, pH 7.8, with the appropriate concentration of thiol and mixed disulfide glutathione added to facilitate oxidative shuffling. And as you see, we see two major peaks that have formed, peak 2 and peak 1. As it turned out, the peak 2 was the correct molecule that folded, and peak 1 turned out to be the D-cis at position 47 that had racemized during the synthetic steps for making the full-length peptide. Following preparative HPLC, we were able to separate out the two components, and as you see here is the HPLC analysis as well as the mass spec, which is consistent with the forming the 4 intramolecular disulfide bonds. The purity you see is around 88%, which was adequate for the biological studies that we were initiating with our collaborators over in Singapore. What's interesting to note here is that the earlier eluting species, the peak 1, also had very similar properties in terms of its ability to give a very nice NMR signal. And it was actually subsequently crystallized, as was the original material in Dr. Chandy's lab over in Singapore, but only the material that was crystallized from peak 2 gave a high resolution X-ray crystallographic structure. What you can see here is our yield table for each of the analogs that was produced. We see that each of the peptides made approximately 0.3 millimole scale. VVK1 had the lowest yield in actuality because we were competing with the D-isomer that had formed as well. When we optimized the synthesis somewhat and changed a few of the conditions for deblocking, mainly adding HOBT into a piperidine mixture to help buffer the piperidine, we're able to improve the overall yield by minimizing that deformation of the cysteine residue. The EGK5, in fact, we were able to further optimize and get the yield up to around 10%. The EGK6, the minimal dimer that we had, the minimal structure, which was similar length to the scorpion toxins, also about a 10% yield, and the TAMRA labeled material also around 8% yield. As I mentioned, we were able to crystallize the BVK peak 1 or peak 2 material. And this particular material gave a highly ordered structure, which compared with the another scorpion toxin called Agitoxin, you can see they give highly homologous three-dimensional structures with the characteristic alpha helix that you see here, and overlaid with the 3 anti-parallel beta pleated sheets. The next real critical feature was to see whether or not these peptides did in fact block Kv1.3. And as we were set up from our long-term collaboration to have electrophysiological tests for this type of work, Dr. Chandy's lab was able to take material and do patch clamping of cells and treating those with EGK, EGK5, and BVK. And in each of the cases, you can see that the peptides didn't really block the channel per se. You can see as a control, 1 nanomolar margatoxin completely suppresses the signal here. And similarly, SORA4 dye, which is a different type of molecule, a small molecule drug, which also inhibits KCA channels as well, also blocks the signal. So what we were able to see is we weren't able to block the channel, and furthermore, there was no competitive interaction with margatoxin. This was somewhat disappointing to us. However, we continued with our additional experiments, and we were able to purify Kv1.3 using a his-tagged channel and using a cobalt affinity column. And what you could see was when we took the peptide and we loaded it with the EGK5 in the presence of the channel loaded material on the column, what we would see is that the peptide basically was not eluting when the peptide was in the presence of Kv1.3. And then when the column was eluted with imidazole, the peptide eluted. Mass signal was observed, and the second elution, we observed that furthermore. And in the case of the EGK6, we saw that it did not bind in any of the channel situations. And in the case where the column had no channel bound, you can see that the EGK5 was basically washed out through elution 1 and 2, small trace amounts present. But clearly our idea that it was interacting with Kv1.3 was substantiated by this data. So we proceeded to look at an additional study, which was basically Biolayer-Interferometric analysis of EGK5 to purified Kv1.3. And what you can observe when you do this type of study is that increasing levels of the toxin, or the plant defensin in this case, in the presence of the Kv1.3, which is immobilized on the sensor tip, we could basically see an increasing dose-dependent interruption of the signal as determined through the biolayer analysis. And when plotted, it showed us that we had an approximate Kv1.3 dissociation constant of 0.52 micromolar. Now, our real goal was additionally to understand whether this peptide, because of the arginine-rich region, could potentially translocate across the cell membrane. And the next set of experiments was in fact to test that idea. And to better understand that, we took the peptide, which had been labeled with the TAMRA, and we measured its ability to enter the plasma membrane or into the cytosol, if you will, using L929 cells, which are expressing Kv1.3. When incubated with the peptide for 1, 3, and 24 hours, the cells were then subsequently washed to remove any of the free TAMRA peptide and stained for actin. to visualize the cellular outline, and Hertz dye to help delineate the nucleus. Using confocal microscopy, the TAMRA EGK5 is primarily seen in the membrane overlapping with the actin staining that you can see here. And what we further saw was that the peptide, in fact, seems to enter the plasma membrane, and a very small portion entering into the cytosol, but mostly located associated with the plasma membrane. It did not alter the viability of any of the cells that were tested, and this was determined by taking the cells and treating them with trypin blue and 7-ADD to measure their ability to exclude these. And we see that the cells remain more than 100% viable, so it wasn't an effect of killing the cells that was causing the material to enter into the cells. It was truly a situation where the peptide was actually passing into the cell using the motif of the arginine-rich region, as we had speculated. We then proceeded to look at the product and test its ability for ameliorating symptoms of rheumatoid arthritis using a model that we had worked on in our laboratory with Dr. Christine Beaton at the Baylor College of Medicine. And she was able to determine that, using rat collagen-induced arthritis, the TEM cell-mediated model for rheumatoid arthritis, Lewis rats are first immunized with collagen and incomplete Freud's adjuvant. They develop arthritis typically within a week after receiving a booster of collagen and the adjuvant emulsion. Treatment is then started at disease onset after the first signs of arthritis, typically swollen joints with a score of about 2. At this time, the rats are placed in either a vehicle or a treatment group, and rats are injected subcutaneously every other day with 0.1 mg per kilogram or with vehicle. The vehicle-treated rats develop clinical scores of approximately 30, indicating moderate level of inflammatory arthritis, whereas rats treated with the EGK5 typically exhibited significantly reduced clinical scores. And furthermore, looking at the X-ray treated, you can see that the healthy rat versus the vehicle, you can see inflammatory responses at the yellow arrows indicated here. And in the situation with EGK5, you can only see very moderate inflammatory responses in the rat X-ray. What we were able to then look at is doing some histological examination. We showed a subset of inflammatory infiltrates at the joint, which with EGK5 treatment, these ameliorated the inflammatory responses as shown here in the two lower panels here. These results suggest that the accumulation in the joints is specifically modulating the T effector memory cells and help to potentially serve as a treatment for rheumatoid arthritis. Furthermore, we did a whole series of tests in a clinical study to look at whether the peptide showed any types of toxicity. And in vivo, at 10 times the effective dose, no toxicity was observed whatsoever. So with that, I just want to conclude this portion of the talk that we've identified some novel Kv1.3 specific plant defensins. These inhibit the Kv1.3 based upon an intracellular domain that they interact with, which in fact is a PIP-dependent mechanism. Didn't really have time to cover that today, but some studies were also in the paper which we published showing this. We designed an optimized analog which had sub-micromolar affinity for Kv1.3, and we were able to show that EGK targets the joints in animal models and ameliorates symptoms in an arthritis model. Just briefly, I want to mention that AmbioPharm is a peptide CDMO. We're set up to handle basically R&D level all the way up to commercial manufacturing of peptides. We have facilities in the USA as well as in Shanghai, China, with capabilities of producing more than 300 kilos on an annual basis. We have 16 manufacturing lines with 1,000 liter solid phase reactor and 5,000 solid phase reactor, solution phase reactor. We have more than 600 chemists, and we're fully FDA regulated and audited at both of our sites. Very recently, we signed a new deal with a company that many of you will know, CEM, which is a leader in developing microwave technology for peptide synthesis. And in fact, we have established the GMP synthesis capabilities using CEM's Liberty Pro at our North Augusta facility in South Carolina, which now adds to our facility the capability of not only doing downstream processing, but fast, efficient, and green microwave chemistry to handle GMP projects, especially those that have critically tight timelines. And we welcome you to explore partnership with us, with CEM, and help to advance your product into clinical development and hopefully ultimately into commercial. With that, I'd like to thank our collaborators, Dr. Chandy at Nanyang, Ray Norton at Monash, Serdar Kusiak at University of Sydney. I led this work while at Peptides International and then carried it forward to AmbioPharm. UC Davis with Heike Wolf's lab, Christine Beaton, as I mentioned, at Baylor College of Medicine, and at Tetra Genetics with Paul Calusi. Thank you again for your time, and I appreciate the opportunity to present our data to you. Have a great rest of your conference.